Prophase 1.
Prophase 1 is the longest of the phases. The long strands of DNA coil up and form chromosomes. The chromosomes are made up of two identical chromatids, which are identical copies of the same DNA strand. The chromosomes are joined in the middle, or the scientific term, centromere. The crossing over point is important because that's where the genetic shuffling takes place. If it was always the same, there would be no difference between the parent and the child. Once crossing over has happened, the centrioles start to pull apart and toward the poles of the cell. This is also where nucleus disappears and the nuclear membrane starts to break apart.
Metaphase 1.
Homologous chromosomes pair up at the middle of the cell, and the spindle fibers catch on to the homologous chromosomes and pull them apart.
Anaphase 1.
Homologous chromosomes separate and get pulled to the opposite poles of the cell by the spindle fibers.
Telophase 1.
One chromosome from each homologous pair is at each pole of the cell, and a nuclear membrane forms around the chromosomes at the poles of the cell.
Interkinesis.
This is the stage between cell divisions. During this time, the cell will grow and make proteins as interphase of mitosis. Unlike interphase in mitosis, there's no copying of DNA during this stage.
Prophase 2.
In prophase 2, the nuclear membrane breaks down and a new spindle forms. There is one chromosome of the two homologous chromosomes in each cell.
Metaphase 2.
In metaphase 2, the X-shaped chromosomes form a single line across the middle of the cell and the spindle fibers bind to both sides of the centromeres.
Anaphase 2.
In anaphase 2, spindle fibers contract and the sister chromatids move towards opposite poles. Once they separate, each sister chromatid is considered to be a chromosome.
Telophase 2.
In telophase 2, nuclear membranes reform around the sets of daughter chromosomes and spindle fibers begin to disappear.
Cytokinesis.
In cytokinesis, the two daughter cells are separated.